Welcome to your custom-tailored deep dive. We are jumping straight into the source materials you provided, and today's focus is incredibly relevant if you're prepping for discussions on the future of clinical genetics or personalized medicine.
Oh, absolutely. It is a very timely subject.
Yeah, we are examining this highly significant 2025 study from BMC Biology by O'Brien and colleagues. And the mission today, our overarching goal here, is to explore how scientists are using C. elegans.
Which are microscopic worms.
Right, microscopic worms. They are using them as patient avatars to map 25 different rare human genetic diseases.
It is a massive undertaking.
It really is. We are going to extract the exact aha moments you need from this paper, skipping the jargon overload so you can see how this creates a huge shortcut for drug repurposing.
And the fundamental problem this paper addresses is that genetic diagnosis right now is incredibly fast and cheap. I mean, we are adding over 100 new genetic disorders to databases like OMIM every single year.
But knowing the gene doesn't mean you have a cure.
Exactly. Roughly 95% of characterized rare diseases have no approved treatments.
95%.
Yeah, it is staggering. And what's fascinating here is that finding the genetic cause doesn't automatically give you a drug target, especially because about 74% of rare diseases affect the central nervous system.
Right, the brain and the spinal cord.
Right. And modeling that level of neurological complexity in traditional models like mice is just way too expensive and slow for a high-throughput drug screen.
Okay, let's unpack this, because the researchers essentially bypassed that whole vertebrate bottleneck using these worms, right?
Yes, they did.
Which sounds crazy because you think, how can a millimeter-long worm model a human brain disease? But C. elegans are perfect for this. They actually share over 3,000 conserved genes associated with human diseases.
It is a surprisingly robust model organism.
But they didn't just knock out a gene and stare at a worm under a microscope. The setup here was this automated, multidimensional tracking infrastructure.
Yes, the methodology is astonishing. They used 96 well microtiger plates.
So 96 tiny little test tubes glued together, essentially.
Basically, yeah. And they paired them with custom high-resolution imaging rigs and a software pipeline called TIRPSY.
TIRPSY. And they put the worms through a very specific 16-minute audition in these plates.
A very synchronized audition. The protocol is strictly controlled.
Right, so first there's a 5-minute pre-stimulus recording.
Just to establish a baseline behavior, yes.
Then the environmental stressor, a 6-minute blue light recording. They deliver 3 distinct 10-second pulses of really high-intensity blue light.
And that is crucial because C. elegans have a very well-documented photophobic escape response.
They hate the light.
They absolutely hate it. It triggers the light-1 receptor, which forces this really rapid, complex motor response they're trying to escape. And then they follow that up with a 5-minute post-stimulus recording to watch the recovery.
To see how they calm down, and the sheer volume of data this generates is it is an explosion of data.
Oh, it really is. The TIRPSY software effectively skeletonizes the worm in the video. It breaks the body down into the head, neck, mid-body, hips, and tail.
And it extracts, what, over 8,000 features?
Exactly. 8,289 behavioral features for every single well.
That is wild.
It tracks the angular velocity of the head, posture, locomotion, morphology. What stands out to you when you look at a data set like that? Because it essentially creates a highly specific behavioral fingerprint for a disease.
Without even needing to fully map the biochemical pathways first.
Exactly. If the mutant worm's behavior mathematically diverges from the healthy wild-type worm, you have your screenable baseline.
So to test this, the first big genetic network they targeted was the BORC complex.
Right, BORC.
Which controls lysosome positioning in cells.
Basically, the trash disposal and recycling centers of the cell. They have to be moved down the long axons of neurons.
And in humans, if the BORC complex is broken, it causes severe stuff. Hermansky-Pudlak syndrome, neurodegenerative diseases.
Yes, and if you completely delete BORC genes in vertebrate models, it is lethal. The embryos just die.
But the worm knockouts lived.
They did. The orthologs BLOS1, BLOS8, BLOS9, and SAM4, the worm survived. And the TIRPSY system found over 3,000 statistically significant behavioral differences from the normal worms.
Here's where it gets really interesting, because the phenotypes weren't all the same across that complex.
No, they weren't.
The BLOS1, BLOS9, and SAM4 mutants, they were all shorter in length. They had decreased head movement.
Right, a very correlated cluster.
But the BLOS8 mutant was the outlier. It actually became longer than a normal worm, and it had an increase in head curvature.
Which perfectly highlights why this system is so powerful. Structurally, the BLOS8 protein is totally unique in that complex.
How so?
It is dispensable for transporting synaptic vesicles, but it's absolutely essential for transporting lysosomes. The other proteins overlap both jobs.
So the software picked up on that exact structural nuance just by watching how the worm wiggled.
Just by the motor output and morphometrics.
Hey,
It is incredible sensitivity.
that is amazing. And they didn't stop at neurodegeneration. They also pivoted to oncology. Bird-Hogg-Dubé syndrome, or BHD.
Yes, driven by mutations in the folliculin gene. It causes things like lung cysts and renal cell carcinomas.
So they knocked out the worm versions of those genes, the FLCN1 and FNP2 genes.
And the behavioral fingerprint there was severe hyperactivity.
They just wouldn't calm down after the blue light.
Exactly, they failed to habituate. But the real utility of the model is testing drug sensitivities. The folliculin complex interacts with cellular metabolism pathways, specifically AMPK and MTOR.
So they threw MTO modulators at them.
Yes, and they found a distinct split. The FLTN1 mutant was much more susceptible to MTORC2 inhibition using a drug called JRAB011.
Well, the FNP2 mutant was sensitive to something else.
Right, it was more susceptible to dual MTOR1 and 2 inhibition using a drug called INK128.
Meaning they can use these worm avatars to find highly specific drug vulnerabilities for rare cancers.
Exactly, it completely streamlines the screening process.
But deleting a whole gene is sort of the easy mode, right? In real patients, diseases are often caused by just one single typo in the DNA, a single amino acid substitution.
Which is incredibly hard to diagnose and treat.
Yeah. The paper brings up the specific case with Cornelia de Lange syndrome. A patient had what they call a variant of uncertain significance, a VUS.
In the SMC3 gene.
Specifically the K114E mutation.
Right. And interpreting a VUS is one of the biggest headaches in medical genetics. You sequence a patient, you find a mutation, but you have no idea if it is the actual cause of the disease or just a harmless quirk in their DNA.
And you can't just knock out the SMC3 gene to test it because complete loss of cohesion, the complex it's part of, is embryonically lethal.
So they didn't knock it out. They used CRISPR to perfectly recreate that exact human patient's typo inside the worm's genome.
The SMC3 K115E worm.
Exactly. And because it was just a structural tweak, not a deletion, the worms lived. They hatched normally.
But the tracking software caught the problem.
Big time. 5,685 behavioral differences.
Things like abnormal development, increased mid-body curvature, decreased head velocity.
If we connect this to the bigger picture, generating that massive feature profile empirically proves that the patient's specific VUS was pathogenic.
It was the cause of the disease.
Yes. It ends the diagnostic odyssey for the family right there.
And instantly gives them an avatar to start testing drugs on.
Precisely.
But it's not always that easy. The paper brings up this final hurdle, the TNPO2 mutation.
The MB2 gene in the worm.
Right. It causes a super severe, ultra-rare neurodevelopmental disease in humans. So they made a patient avatar for the specific typo, D157N, but the worm barely showed a phenotype.
It only had 770 feature differences from the wild type.
Which sounds like a lot, but for a high-throughput drug screen, that is basically useless.
It is entirely useless. It's a cryptic phenotype. Once you start dumping thousands of chemical compounds into those microtiter plates, the background noise will completely mask a signal that week.
You'd never know if a drug was actually working.
Exactly. The biological defect is there, but the standard test isn't stressing the worm enough to make it obvious.
So the researchers had to coax it out of hiding?
They tried a few things: temperature stress, RNA interference, but chemical sensitization was the key.
They used a drug called aldacarb.
Yes. Aldacarb stops the breakdown of acetylcholine in the synaptic cleft.
Which means the chemical messages between neurons just build up and build up.
Until it becomes toxic. In a normal, wild-type worm, aldacarb exposure causes hypercontraction and total paralysis within a few hours.
But the mutant worms didn't paralyze.
They exhibited distinct resistance. The normal worms froze, but the mutant avatars kept moving.
So what does this all mean?
It means they found a way to force the cryptic phenotype to reveal itself. By applying that chemical stressor, they transformed a weak, invisible signal into a binary state, paralyzed versus moving.
Which is brilliant, because now you can use that weak model for a high-throughput drug screen.
Exactly.
Right.
You just dump your drug library onto the aldocarb-resistant mutant worms, and you look for any drug that makes them paralyze again.
A drug that reverses the resistance.
Right. It exposes the hidden
That
vulnerability.
is such an elegant solution. Let's synthesize the takeaways for everyone listening. We've gone from automated cameras tracking thousands of microscopic worm movements all the way to finding bespoke drug targets for rare diseases that big pharma usually ignores.
It really is a paradigm shift.
The efficiency here is staggering. They can capture the effects of everything from a full gene deletion to a single amino acid swap in a matter of weeks.
And it shows why critical thinking is so essential in this era of genomic information overload. By standardizing the complex multidimensional behavior of a simple organism, we can systematically map the unbelievably complex genotype-phenotype space of human diseases. This raises an important question, though.
Which is?
How quickly can we scale this?
And that is exactly the final thought I want to leave you with today. Think about this trajectory. If a single amino acid tweak in your DNA can be perfectly mirrored in a millimeter-long nematode to test thousands of existing drugs right now, what happens when we sequence the genome of every single person on Earth?
The logistics of that are fascinating.
Will we eventually have a massive library of worm avatars for every human being just waiting on a server somewhere to test your specific illnesses before you ever take a single pill?
A fully operational, personalized medicine pipeline.
It is wild to think about. Thank you so much for joining us for this deep dive into the source material. We hope this gave you exactly the insights you're looking for. Keep questioning and keep exploring.